Pharma 4.0. What is the meaning of Pharma 4.0? This is the application of Industry 4.0 technology, applied application of artificial intelligence, the Internet of Things, IoT, and the digital twins to pharma manufacturing and development for improving the production efficacy, quality assurance, and regulatory compliance guidelines. Then this is the steps involved in the syllabus. First one is the foundation, introduction to chemometrics and multivariant data analysis. Second step, concern for the spectroscopic data modeling. Third stage, quantitative analysis using the various regression analysis techniques. Fourth one is separation sciences, chromatographic peak modeling. And fifth one is concern for the quality and security for using the models to identify genuine versus counterfeit detection. The sixth step is signal integrity, how to reduce the noise and get a definite peak of the either a definite result with the separation of several variabilities. And seventh one is governance, that means that limitations of AV and what are the models are used, but what is the major limitations of artificial intelligence in pharma analysis. Means that each module is not an isolated unit, that is the stepping stone towards the realizing quality by design and process analytical technology. This is the present concept running in the research, quality by design and process analytical technology to develop the aims and objectives of QBD and PAT. We should use the models of artificial intelligence by using the respective steps involved in the process. Then coming to the topic 1, the multivariant advantage and the matrix architecture. The syllabus outcome is students will master the data matrix and they will learn to extract redundant informative signals from noise using latent variables and principal component analysis. Previously, the SWAN world coins the first term, the name is chemometry, to apply statistical logic to chemical data. Presently, adopting the PCA and parafact analysis for continuous process monitoring. What is the meaning of parafact analysis? It is the parallel factor analysis. It is one of the mathematical process used in the chemometrics and multidimensional data analysis. What is the feature of the syllabus given here? The integration of multivariant data directly into automated real-time release testing framework. That means we want to analyze the data available in a very speedy way. Then, what are the reference books or literature available in the today's market? First one is Handbook of Chemometrics and Qualimetrics. The role of the syllabus is the definitive blue book providing the exhaustive mathematical baseline for creating chemical measurements as multivariate spaces. Second book is Chemometrics, a practical guide, written by B. Fell Sishalt. The role of the syllabus is making the bridge gap between the abstract multivariate statistics and applied laboratory workflows. Then the third book is a multivariate statistics, a practical approach, published by Fleury and Redville. The role of the syllabus is it offers highly accessible introductions to complex data analysis for students without deep statistical background. Even though the students who are not know mathematics very well, by using these techniques, we can able to complete the practical work in our laboratories. This is the overview of the topic, chemometrics and multivariate analytical data. The chemometrics uses mathematical and statistical methods to extract the useful information from chemical data. Then the first point is chemometrics that includes the mathematics, computer statistics, and computer sciences. It helps to design experiments, analyze data, and interpret results to make reliable decisions. Second one is purpose in pharmacy. What is the purpose of chemometrics and multivariate analytical data? It is used in different areas of pharmacy to deal with complex data and improving the decision making. Thereby, there are several applications of pharmacy. That means for taking the better decision in development, quality control, and quality assurance. By using the multivariate analytical data, you can able to use more than one variable measured simultaneously for each sample and observation. That means in case of EV visible spectrum recording, various absorbances at various different wavelengths and various concentrations at different wavelengths, you can able to study in one step of the process. Common chemometric techniques applied in this practical approach is principal component analysis, that is the PCA model. Second one, partial least square analysis, PLS model, and cluster analysis, HCC. What is the meaning of HCA? That is the hierarchical cluster analysis. That means the software divide the samples into the groups on the basis of their similarity and the distance. The group of one parameter, if you given for the software, that parameter, similar parameter containing compounds are separated as one group and other compounds will be divided into the another group. That is called as the clustering analysis. What is the applications of pharmacy by the chemometrics and multivariate analytical data? First one is formulation optimization. Second one is quality control and its finalization, means that to ensure the product quality. Third one is impurity profiling. Then fourth one is process monitoring during the formulation time. Generally, there are several steps are involved. Then which step is going to be in wrong way, which step is going in the good way, that will be predicted by using the chemometrics and multivariate analysis. The fifth one is method development and validation. Then finally, the data interpretation and decision making will be obtained, thereby convert the complex data into useful information for better decisions. Shortly said that chemometric transforms complex multivariate data into meaningful information, improving the efficiency, accuracy, and reliability in pharmaceutical research, development, and quality assurance. Then coming to the topic 2, spectroscopic data modeling and molecular characterization. This is one of the important thing happening in our practical approach. It is mostly useful in our B.Pharm level also especially. What is the outcome of the syllabus is students will learn to translate EV visible, IR, NIR, and Raman light matter interactions into structural insights, mapping electronic transitions and vibrational modes to chemical composition. Generally, the mapping of the electronic transitions and vibrational modes, we can't able to visualize what is happening in the instrumentation. In case of the UV visible and IR spectroscopy, that will be studied by using a new software developed. This is what is the past condition of this technique. It exclusively on physical reference libraries or computationally exhaustive quantum mechanical AB initio calculations. What is the meaning of AB initio calculations? That is the from first principle. AB initio means the first principle. What is the meaning of first principle in terms of the molecular? That means that how the molecular structure will be developed how the bonds will be developed, what are the energy levels of each and every bond, and what nature of the chemical bonds are available. On the basis of that bonding nature, the the data will be obtained from the structure. In the future, the next coming days, the science using the deep learning architecture, which is called as EV visible scanner and MPNN, while predicting the spectral signatures and phototoxicity directly from 3D molecular coordinates with RMSE values, calculating the RMSE values as low as 0.06. What is the meaning of UV visible scanner? The UV visible scanner is nothing but deep learning model predicting the absorbance of molecules. That means we can able to predict the UV visible spectra. What is the meaning of MPNF? Message passing neural network. Message passing neural network. That means that molecule formation takes as a graph and predicting the properties of the molecule. How to make the graph from the molecule formation on the basis of atoms, on the basis of bonds formed in the molecule. That MPNN software predicts the graph from the molecule formation and gives the molecule properties. Finally, you will get the predicted either V visible spectra or IR spectra or NIR spectra or Raman spectra. Then, what are the sources of this material? First one is Handbook of Near Infrared Analysis, published by Workman and Suyarjak. The role of the syllabus is the authoritative test on extracting structural data from the highly convoluted overtones and combination vibrations in the near infrared region. The second textbook preferred is Modern Raman Spectroscopy, a practical approach, published by Smith and Dent. The role of the syllabus is provides the mechanical and theoretical ground for dispersive Raman, FT Raman, and surface enhanced Raman scattering phenomena. Then the third book suggested is Machine Learning in Quantum Calculations. This will be preferred by the literature survey in the Google search or any other website. The role of the syllabus is curated recent papers demonstrating the training of EV visible transformer and MPNN models against the quantum mechanical benchmarks for high throughput drug screening. This is the summary of the spectroscopic data modeling of the UV IR interpretation. The first 3 points all are well known. What is the spectroscopy, what is the UV visible spectroscopy and its electronic transitions, what is the IR spectroscopy and its vibrational transitions. And now, by using the multivariate analysis in spectroscopy, the overlying spectra are converting into the multivariate modeling spectra, which are individually recorded. Thereby, spectral data contain overlapping peaks and noise. That overlapping peaks and noise will be solved by using the multivariate techniques help to extract useful information. The general models used is PCA score plot and the PLC model and cluster analysis and the HCA techniques. These techniques already shown myself in the previous slides. This is the one of the importance of the model is PCA analysis one, that is the principal component analysis. See in that plot, you can able to identify that a green, blue, and red color spots. That means that how the sample will be grouped. If the sample is the mixture of the component, then the sample of the molecules will be grouped into the different spots, and you can able to predict and identify the which sample and what group it will be, what is the quantitative of the respective group of the molecules present in the given mixture. The benefit of this type of the models are noise reduction, pattern recognition, classification, quantification of the mixers. Then coming to the applications in pharmacy by using the spectroscopic data modeling is first one is drug identification, purity testing, quantitative estimation by using the calibration models, and impurity detection using the identify and quantify trace impurities in the formulations. And the fifth application is the quality control system to ensure the batch to batch consistency and regulatory compliance. It is the most important task. Several batches are running, each batch several steps are included, thereby batch to batch consistency smoothly going or not. We can able to study it by using this new software spectroscopy data modeling techniques. Similarly, once the batch to batch consistency is identified, if the batches are passed automatically, they compliance with the regulatory guidelines. This so the spectroscopic data modeling converts raw spectral data into meaningful chemical information, aiding accurate identification, quantification, and quality assurance in the pharmaceuticals. This is the step two. Then coming to the topic three, quantitative regression models, PCR versus PLS, where using the two systems, one is the principal component regression analysis, second one is the partial least square analysis. What is the first one? Previously, we are used for rigid global multiple linear regression analysis and basic PCR facing failure under severe multicollinearity. So previously, lot of failures are there because of we are using the physical methods. Presently, the scientists are applied integration of artificial neural networks and support vector regression to manage highly nonlinear QSAR or QSPR models. QSAR chemistry peoples are well known. What is about on the QSPR? The QSPR means the quantitative structure property regression model. Quantitative structure property regression model used in the chemoinformatics and computational chemistry. Then where you will get the literature from the above said topic. The first material, the first subject on the textbook is multivariate calibration, published by Martens and Nice. The role of the syllabus is the classical rigorous mathematical foundation for PLS regression and its application in industrial settings. The second book available is a user-friendly guide to multivariate calibration proposed by Nas, Wisconsin, Farrell, and Davis. The role of the syllabus is translates dense calibration mathematics into actionable workflows for laboratory practitioners. The syllabus role is advanced readings on using PLS and ANNS to predict pharmacokinetic behavior, placental barrier permeability, and drug resolution profiles for these experiments. This literature survey is required from which we can able to get the practical exposure. Then the summary of the respective regression analysis used in the quantitative pharmaceutical analysis is definition. Regression analysis is a statistical tool used to study the relationship between a dependent variable and independent variable and to use this relation for the estimation of unknown concentrations. Then this is all a known one. Calibration, that is a linear regression, calculation of the regression parameters, and goodness of fitting. If you will get R square value close to 1, indicates the linearity is successful. That means the working model is the successful. Then here the given one example for how to find out the unknown concentration from the given data set. Then 4, 5, 6 points are explained. The concentration of unknown sample gives the 7.01 microgram per ml on the base of y is equal to mx plus a calculation. Now we have to apply the software to identify the goodness of fitting. See the 7-point residual plot diagram, which explains randomly scattered around the zero line means that the good fit of the model. Otherwise, the model is not correct one. We have to go for the next trial. What are the applications in pharmaceutical analysis? The standard solutions are prepared, that is all are now known. Then measurement by the UV visible or HPLC, construct the calibration curve, and you can able to analyze the unknown sample concentration used for the quantification of drug concentration in the given unknown sample. The chromatographic peak modeling and AI deconvolution, that is the topic 4. It gives the good demonstration of the working condition, where you can see in the past constraint and the manual integration, there is a lot of clustering is there. In the chromatograms, there is a lot of cluster is there. But by using the AI deconvolution model, then the peaks are separated well. The compound peaks are separated well. Means that in case of the past working condition, rule-based mathematical derivative logarithms prone to error under matrix effects, highly vulnerable to regulatory scrutiny. But by using the one-dimensional convolutional network, neural network that learn spatial peak patterns paired with graph neural networks for precise covariance retention index predictions. Then where that material is available? The material is available in the described 3 textbooks, 2 textbooks, and some of the material we want to gain from the literature source. The first one is multi-way analysis literature, Bro and Case. The syllabus role is core reading on Parapath 2, a critical direct fitting algorithm specifically designed to model chromatographic data experiencing retention time shifts. Then the second book proposed by Professor Vatkosar, The Chemometrics in Analytical Chemistry. The role of syllabus covers the optimization of mobile phase compositions, flow rates, and gradient profiles using statistical experimental design. Machine learning in chromatography is the derived from the literature. It should be derived from the literature. The role of the literature is contemporary papers on deploying support vector and random forests to optimize the compounds identification from the complex sample matrices. This is the brief discussion about on the chromatographic peak modeling and interpretation. The first 6 points all are known one: ideal Gaussian peak, peak parameters, peak symmetry, resolution, peak area, and overlay of the peaks which are co-eluted. Then, if the overlay of peaks are obtained from the chromatography, how to separate into individual peaks of the compounds? By using the chromatographic modeling software. By using this software system, using the application of AI, we can able to separate the mixture of the chromatograms into the individual chromatograms, which has been shown in the figure number 7. Which has been shown in the figure number 7. See here, T1, T2, T3, T4. 4 compounds are separated on the base of their retention time. Then, by using these models, mathematical models, we can able to measure several parameters. Similarly, coming to the applications of this topic is identification of the compounds on the base of the retention type, quantification of the drugs and impurities by area-wise, area that means a peak area. Third one is purity assessment and impurity profiling and method development and optimization. The final application is quality control and regulatory compliance. Then coming to the fifth topic in the syllabus is classification of models for genuine versus counterfeit detection. The today's software using is the Raman barcode translator, where we can able to predict the given prediction is the pure or the given sample is pure or not, either API or formulation. What is the outcome of the syllabus is authenticating API. Spectra are transformed into discrete barcodes, support the vector machines, which classifies the overlap to detect falsified medicines with near 100% accuracy. Previously, what is the happening? It is the destructive process we use, time-consuming laboratory HPLC testing, resulting in supply chain bottlenecks. But in case of the future, you may get the benefit from this artificial intelligence neural network system. Handheld spatially offset Raman spectroscopy applied system, that is the SORS, paired with cloud-based ML machine learning, enabling real-time through packaging authentication in the field. The syllabus available in the prescribed textbooks, one is Human Metrics for Pattern Recognition proposed by Brereton and Forensic Applications of Raman Spectroscopy by the others was suitable others, selected the others. That is a little bit difficult to obtain this textbook. The role of the syllabus mentioned here is specialized literature on the chemical profiling of medicine counterfeits, linking spectral data to organized crime forensic intelligence. Then the Raman barcode system should be identified from the selected literature where you will get the practical exposure. How to separate the genuine sample versus counterfeit detection? In the first point, you can observe here the prediction is genuine or counterfeit from the given data by using the data pre-processing step followed by the classification of the model. In the second point, you can observe here data in feature space. The blue dots are genuine and red spots are counterfeit samples. Then decision has been taken here. Blue spots are genuine and the red spots are then makes the one zone. Then by using the model training and prediction, we can able to predict the given unknown sample is good or counterfeit sample. The model is evaluating using the test or validated data. Then we can able to predict the given sample is the genuine or counterfeit sample. By using this type of the calculation by confusion matrix model, we can able to calculate the accuracy as well as sensitivity, specificity, precision, and the other scores required from the respective the data set. The common classification models used are here LDA, linear discriminant of analysis, SVM, support vector mission, and KNN, K nearest neighbor, and random forest techniques. These are the four models are generally used for using the artificial intelligence neural network system. Thereby, you can see in the seventh point, there the system predicted the given sample from the given spectra. The software designed as classified as genuine for the given sample. Then overall workflow of the system is collect the data and give the data to the processing. Then model will be trained to detect to take the decision and validate the model. Finally, predict the new sample. Then this data, this workflow use the help in quality control, regulatory compliance, patient safety, and combating the counterfeit medicine. Then coming to the applications of the syllabus is detection of counterfeit drugs in supply chain, quality control in pharmaceutical industry, authentication of raw materials and finished products, regulatory agents and forensic investigations, and useful for protecting the public health and builds trust in the medicine. Then last but one topic, handling noise, scattering and signal variability. This is one of the difficulty of the practical work to reduce the noise and improving the signal processing. If you are using the AI technique, it will be easy to carry out. You can see here, in case of the past, the using the Savitsky-Golay transformation system, you will get the peak, but it is very broad one. But in case of the physics-informed deep learning software system, the peak will be very definitely obtained. That is the advantage of the a artificial intelligence software model. in case of the noise reducing and signal recording system. The syllabus overviewed in the textbooks are data processing practice strategy in ATR FTIR proposed by Lee et al. Deep learning for reconstructing low quality spectra proposed by Brodant et al. And peak clipping algorithms for background estimation proposed by Morak and Matosco professors. Then, for giving a summary about on this topic is you can observe here, observe in the first point, there is a lot of noise is there along with the peak. Then noise reduction has been takes place. Finally, there is the baseline correction takes place and we will get the neat chromatogram after well separation of the peaks. Then by applying the respective artificial intelligence system, you can here the spots are spreaded, means that the spreads of points shows variability in reapplicate measurements due to random error. These are very close to each other. If they are far near, far from distance to each other, means that your trial will be failed. Similarly, total data sets will be take the decision and measured values are this take the calculation for which gives the measured value from the 5 to 6 samples. You can take the average, then you will get the correct decision about the chromatogram obtained is correct in mode or not. That means that averaging replicate measurements gives a more reliable result and reduces the random error. Then the models used in case of the noise and variability is baseline correction, normalization system, statistical or chemometric methods, either PCA, area under the curve, and standard deviation models. Otherwise, averaging the reapplicate measurements means that the fifth point which we have discussed just now. And other one, filtering system using the Fourier transform system or wavelet system. Otherwise, the smoothing system that is the using Savitsky-Golay software system. What is the importance in pharmaceutical analysis? It improves the accuracy, precision, and reproducibility of analytical results, and also reliable for the quantification of drug assays, analysis of the impurities, and the stability studies, and also helps in better decision making, in quality control and method validation. So here you can observe in the bottom of the slide, you have once you have given the raw data, it pre-processing the data and clean the data where you will get the less variability and obtain the a reliable information and get the a neat and defined peak of the chromatogram. Then coming to the limitations of AI and the drive for explainability, even though having this much performance efficiency of the artificial intelligence system, presently it suffers from 9 limitations or drawbacks in its working condition that we are going to discuss in the next slides. The syllabus outcome is addressing the core risks of artificial intelligence in pharmaceuticals using the dataset bias, severe overfittings, and the lack of mechanistic understanding in purely data-driven models. In case of regulatory evolution in future, it is moving from opaque end-to-end neural networks facing regulatory rejection towards the explainable artificial intelligence model. By implementing sharp values to highlight precisely which chemical or spectral region drive a model's prediction, aligning with the MHRA AI airlock program system. What is the meaning of sharp? The sharp is the each individual contributor helps to make a model prediction. Sharp explains that it gives a values explains the contribution of each feature to a model's production. What is the meaning of MHRA? That is the medicines and healthcare products and regulatory agencies which develops AI-based medicine technology useful to control the regulations. Useful to control the regulations. By using this as a sharp technique and MHRA technique, we can able to produce the a benefit more produce the valuable medicine with efficacy, safety, and which is the better health benefits of the medicine. Then this is the literature available by limitations of artificial intelligence. The elements of statistical learning proposed by Hasty, Ifsharani, Friedman, and interpreting the model predictions with the sharp proposal published by Landberg and Lee, and regulatory guidance on AI in healthcare system, that is for the MHRA FDA literature. We have to take from the directly from MHRA and FDA sites. You will get the what are the limitations of the artificial intelligence. Then coming to the first limitation of artificial intelligence, that majorly depends on data quality. If we have given the pure data of our non-pure data, it gives that prediction is incorrect one. That's why AI models are only as good as the data they are trained on. Noisy, incomplete, or biased data leads to the incorrect prediction. So we have given the pure quality data, the poor quality data, we are getting the unreliable prediction. That is the one major limitation. The second major limitation is that lack of transparency, which act as the black box. That means if something is happening, either good or bad in the result prediction, it can't give how it will be good and it can't justify how it is the bad. That is the difference between AI to the our human beings. The human beings, suppose we are working in the one experimentation. If we fail, we have the justification. If successful, we have the reason to successful. But in AI model, it is difficult to get. It is not possible to get the data why it is wrong, why it is successful. That's why the scientists have said that many AI models using the deep learning techniques work as the black box. That means that difficulty to understand how the model arrives at a particular decision. That means internal reasoning not visible. That gives whatever the thing it has the capacity, then it gives the output and decision, but not able to give the reason why the decision has been taken. Similarly, the third limitation is limited generalization. That means that high performance on known data and poor performance on the new data. Then, if that depends upon the known data means that always known data is not available. Then how to guess the prediction? That is the another limitation of the AI in the pharmaceutical sciences, especially analysis. Then fourth limitation is need for large training data. Most AI models require large amounts of labeled data. In analytical chemistry and pharma, high quality labeled data may be limited or expensive to also obtain. In case of general laboratories, it is difficulty to get the large data, training data. That's why the small data set, model underperformance, and large diverse data set, a better results will be obtained. That means that for this software, very huge data is required. That huge data may be available, may not be available for the working condition of the practical or either the work in progress. Then other one is the sensitive to bias. The biased or unrepresentative training data leads to a biased decision. It takes the biased decision. It is not correct in the pharmaceutical sciences because of we are producing the health benefit products. Can result in unfair or unsafe outcomes in critical applications. That means the critical applications it gives the unfair and unsafe applications. That's why the sensitivity to the biased data. Then the sixth limitation is high computational requirements. That means that training complex AI models requires high computational power and time. That means that a best training personnel is required, the software is required, hardware is also required. that may not be feasible for all labs or real-time applications. That is only industry may be possible in lab. In case of academics, it is not possible. So high cost hardware, software energy, thereby not always practically for the routine use. Then the seventh major limitation is lack of domain knowledge. That is the difference between AI, AI knowledge to our human beings knowledge. AI may miss important chemical or analytical context, thereby a purely data-driven models may contradict established scientific knowledge. Thereby, data-driven result may not equal to always scientifically correct. You can't say that it is scientifically correct. Hence, the expert knowledge is still essential. That means our knowledge, our working presence is compulsory required. The eighth limitation is difficulty in validation and regulation. AI models are harder to validate than traditional methods because the regulatory acceptance is still evolving, especially in pharma and clinical labs. That means that complex validation process requires regulatory hurdles and compliance challenges. Recently, I studied one article published by the US FDA peoples. They are also not accepting the AI work, the AI technology completely based on the AI technology. Compulsory hours working experience, hours knowledge, expert knowledge is also compulsory required because of it is missing some of the important concepts while validating the process. Because all are known that the validation process is very tedious. Because of the tedious process, AI may not be validated completely. It may miss one or two points where you fail to get the regulatory acceptance or regulatory complaints of the batch analysis. The ninth major limitation is risk of over-reliance. That is means the over-reliance on AI may reduce the human judgment. That is difficulty to take the judgment by the human, which is correct, which is not correct. Critical errors may occur if a model fails but is trusted blindly. Otherwise, if you do, depending on the AI completely, you want to trust it blindly. It is not correct one. In case of the especially health science, it never trust blindly. You should practically prove that is it right or wrong should be proved. Then only you can get the correct. You can come to the one conclusion. Hence, AI is a tool, not a replacement. Human oversight is crucial. Human must be observed the what the AI is doing, is it reliable or not, should be verified. Simply says that the limitations of AI models in pharmaceutical sciences, AI models enhance analytical decision making, but their limitations must be understood. We should know what are the limitations, where it is going to be paid, where you want to use our expert knowledge and all these things. Thereby, combining AI with expert knowledge, combining artificial intelligence with expert knowledge, quality data, and proper validation ensures reliable and safe outcomes. Means that you never directly depends upon the artificial intelligence and its based software. So this is a feature design PharmaO, the autonomous pharmaceutical analysis laboratory. So this is the new design proposed by the artificial intelligence, the next coming laboratory systems. Their four pillars are there, 1, 2, 3, 4. Those pillars are means that pillar 1 required for data architecture. The syllabus in the syllabus we have discussed the topics 1 and 2. Pillar 2 for the signal integrity, that means topics 4 and 6. Then pillar 3 indicates the predictive modeling. It concerns for the topic 3 and 5. And the limitations of the AI, it concerns for the topic 7. It concerns for the topic 7. This is a feature the pharmaceutical analysis laboratory developed in case of the next future generation. Finally, I would like to say that the syllabus does not just teach algorithms, it engineers the blueprint for continuous real-time quality by design. This is the references I would like to summarize once again. If anybody missed in the previous slides, then see here. The first one is Miller JN and Miller JC chemometrics by the system, and the Brereton RG chemometrics and Messerter DL et al. L. Gindy had GM and BN Colele and Marnoyer, the presently available books in the literature. This is the matching the syllabus. The topic on introduction to chemometrics, better references, Ottosar and Miller proposed books, UVIR spectrum modeling, Brereton and Otto proposed books, and regression analysis by Miller and Miller. The fourth topic, chromatographic peak interpretation by Massart and Ottosar book, and counterfeit drug detection versus genuine samples, Brereton and Bian Colello textbooks, noise and variability handling, and AI limitations by Otto and EI Jindy review textbooks. Thank you one and all for making the patience. So here I have given a brief summary about on the Unit 5 of the AI in Pharmaceutical Analysis and Chemometrics. The course code is BP604P. The subject name is AI Applications in Pharma Sciences. Thank you once again.
Thank you, sir, for providing valuable information related to the usage of AI tools in pharmaceutical analysis. You already given brief information what are different books useful to implement the NEP 2020 syllabus in 601 course. Thank you, sir. Next session by Dr. Bapi Raju. He is going to explain about Pharma 4.0.
So very good afternoon to everyone. So myself, Dr. Bapi Raju from Department of Pharmacy Practice. Thanks for the organizers for this opportunity. So today I'll be sharing my views on one of the newly introduced topic in the BPharm curriculum, which is futuristic pharma through augmented reality and virtual reality, Pharma 4.0. So it's basically a AEC program, which is ability enhancement course, ability AEC course, and we'll be seeing how we can integrate or how we can enable our students with this skill set and how it is useful for them exactly when they move into the industry, Pharma 4.0. So with this introduction, I just go into the slide deck. So I think I'm clear with my screen. So this specific topic, which is 8060, which comes in the 8th semester, which is one of the ability enhancement course, which is an elective, which talks on how we can integrate AR VR in the present field, in the present era of Pharma 4.0. So things before we need to know, like just we need to recollect things before we start is about 4.0. So basically pharma, we have seen industrial revolutions 1, 2, 3, and now we are in the industrial revolution 4, which exactly the pharma industry is also on. So basically pharma 4.0 integrates things like automation, artificial intelligence, IoT, big data analytics, cloud computing, and all these things along with our AR VR technology. And all these are being utilized or integrated mainly for the purpose of improving the process manufacturing, research, quality assurance, and also for the healthcare delivery. So basically, Pharma 4.0 enables intelligent, it connects, and also uses data-driven pharmaceutical systems, which is exactly as required by the Industry 4.0 revolution. So there's a brief overview on how the industry have moved today from its starting 1.0, where mechanization was a primary thing we have used in 1.0, and which next it converted into 2.0, where electricity and mass production has been majorly observed. And next in industry 3.0, computers and automation came into existence. And now we are in the industry 4.0, where all these IoT, AI, and robotics playing major role in the manufacturing process. And the same in case of Pharma 4.0 also, the same we are in that era. So basically, pharmaceutical industries are now entering into an era of intelligent manufacturing and immersive learning. So now we need to make sure that students are acquired with all the skill set which Pharma 4.0 is required. So before we just move, I'll have a small video on how today pharmaceutical industry exactly. And this is one of the video we have from our healthcare. So you can see how robotics are used in the manufacturing process. So this is all happening because of the robotics, artificial intelligence, and IoT. And this is how the present pharma industry is also working on. Starting from detection of defective pieces till the packing, we are using sensors and all these things. So this is how we are using robots which are working on the technology in the pharma industry as well. The technologies, if you look what technologies we are using now in the industry, is industry are using AI ML models starting from drug discovery and development, and it is using IoT sensors for real-time monitoring. So it's in the era of 2.0 and 3.0, most of the things went like human monitoring, but with the advancement today we have, through these IoT sensors, we can easily detect the mislabeled things or defect things in the pharmaceutical industry. And also we are using big data mainly for the process optimization. Today we can do that only because of proper analytics that we can perform with the data that we have. And cloud computing, AR, VR for training and stimulations, digital twins mainly for the training up the freshers. We use these digital twins. And as we have seen in the previous video also, automated things we can do with this particular robots. So these are the main technologies that Pharma 4.0 have been utilizing in various applications that they do on a daily basis. So in this, the main focus that we'll have on today talk is like AR and VR. So before we just go into the details, let's stop here and try to understand what is AR and what is VR. So basically, AR is simple thing that we use in our mobiles. So Google have given an AR cam if somebody have used it. So we can create a lion and we can take a picture of a lion on our bed or on our floor. So it's basically an augmented reality thing. So it combines real and virtual objects. On the real floor, you can create the objects and you can use that. So it is an interactive in real time, and it can be viewed on your smartphones or tablets. And in purpose, if you want to use glasses also, we have specific AR glasses that we can use. So examples that we have is like scanning a medical box for instructions, or AR anatomy apps and Pokemon games and all these things. They are working on the AR models. And coming to the VR models, it's something completely different than AR. So VR basically creates something where AR utilizes the things for the better understanding. So VR creates a complete digital environment, which is a completely computer generated environment, and the user interaction inside a virtual world. And basically, it requires a VR headset to experience that. So today we have various examples and various working models on virtual pharmacy labs, manufacturing training labs, and patient counseling systems, which we have. And in simple words, if you want to know about AR, VR, AR basically enhances the real world, and VR basically creates a new virtual world. It's a basic difference that we have with AR and VR. So here is what we see exactly from this video, what exactly AR and VR looks like. So, so this is a VR you can see in the picture here, in the video here. So on the rear floor, we are putting the things there, and we are trying to see how the place looks. So this all comes under the examples of augmented reality AR. So for the higher performance, basically we use these glasses also, AR glasses, which not at all required, but still you can use that glasses for some better experience that we can have. So the latest glass, if you know, the Apple Vision basically works on this augmented reality thing. And Google Glasses also we have, which works on this augmented reality thing. So, so coming to the virtual reality, obviously we create the things, we create the places in virtual reality. So you can be at your home and you can create some pharma manufacturing unit at your place itself with this virtual reality. And today, even technology has very feasible or friendly, you can say, that we can create AR and VR labs and VR videos for better understanding for the students. So this is the basic difference of AR and VR. So now coming to the applications that we have in pharma education, very particularly with AR and VR, which is driving the pharma 4.0. So we have its various applications in academic learning. We can have 3D visualizations of data structures, and also we can have some virtual models of physiology and anatomy, and also interactive pharmacology learning, which we are mostly using them. And even in labs also, we can create some stimulated settings of compounding lab or machine learning labs, or even you can create some pilot plans using this AR VR technology at the facility that we have. And even that we can extend in the clinical applications also. In example, in the patient counseling simulations or to create a hospital scenario. So we can use this AR VR technologies in creating all these things and improving the performance of the students. And basically, AR VR transforms passive learning into active experimental learning. So coming to the applications of VR in pharmaceutical training in industries or in setups where we require training for the freshers. So we use VR-based training setups basically for the clean room behavior simulations, and we can also train the freshers on GAP compliance, and also we can uh, even the practice on operational equipments also can be done using this VR-based training. And coming to the AR-based assistance in pharmaceutical training or pharmaceutical industry, we can use in SOP overlays on missions or real-time maintenance guidance. So QR-based systems we can create using this ER-based assistance in the pharmaceutical training. Maybe in the patient counseling or patient-centric learnings, maybe VR-based training we can help mental health therapies. We can improve the patient pain distraction therapies using these VR sets, and also rehabilitation support. Also, we can provide using this virtual reality based trainings. And whereas augmented reality in patient-centric learning, maybe we can use them to instruct certain procedures like healing usage, inhaler usage. We can practice them, or we can provide instructions using the AR based setups or tools. And also we can advise them on how we can take insulin or injectable administration procedures or guidelines. And also we can provide some informations on medicine, and all these things we can do easily using your ER based assistance. So we have various wide variety of applications both in pharma education, pharmaceutical industries, and also patient centric learning using this ER and VR setups. So it's very important for today's students to know the basics of AR VR, and also they should be equipped with technology, how they can develop some tools using AR and VR, because this is the need of the hour in the pharma 4.0. So this is how the future of VR in the pharma education is going to be. So we are going to have metaverse classrooms in the future, very coming soon. And also we are going to have remote stimulation labs where uh, uh, even the patient health or patient lab reports can be monitored remotely. And we can have digital twins in pharma, which prevents lot of training schedules for the industry. And also virtual clinical trials are again which is again a broader area that we can expect AR VR influence. And also AI powered virtual tutors also are going to increase in the nearby future in the VR AR systems. So this is how in the next 2050 we are going to see AR VR in the pharma education. So now we are sure that it's very important and need of the hour to learn about AR VR fundamentals in the pharma 4.0. So with that, we go into the syllabus and we'll see what exactly we have and how we can drive the process of learning for the students in this particular aspect. So this 8.806T, which comes under an elective again with the core objectives, like we have 5 important objectives for this course. And first thing is to understand the basics of AR VR technologies and how we can use them in pharmacy, and to learn how to use tools, especially AR VR tools that we use in pharma training and manufacturing, and also to use immense technologies for learning and research activities, and also to know the role of AR, IoT, AI in pharma 4.0, and try to understand the digital technologies that are used in the pharmaceutical industry. So in this, we'll try to cover to the extent possible, and we'll have a roadmap on how we can learn these things in making the students clear about this. Yeah. So here are examples where we can use different tools, which are again open access tools. We can use them and we can train the students and we can train ourselves in this. For example, in the manufacturing training process, we have Siemens Digital Twin Lab. So we can train students on virtual GMP plant training. Maybe anatomy physiological training. We have various VR models on human anatomy. VR is one thing and complete anatomy is one thing. So we can use them in interactive 3D learning models. And we have special labs on AR inhaler tutorials and web AR apps. So we have the apps both in the mobile apps and the web-based apps, where we can use them in demonstrating medical usage. And one more jig space is one more AR-based tool which is basically used in the industries for SOP and equipment training. So we have various tools that we use in industry, and where since they have been open access, we can use them to learn the students how they work in the real world. So coming to the applications in manufacturing, if you see, we can create a virtual GMP plant walkthrough. We can create that using the VR model, AR model. We can train the freshers or people working in the industry on clean room and equipments. We can have some SOP guidelines using the AR overlays. We can create some digital twin based simulation models, and also we can have monitoring of smart sensors in the real time, so that we can have some process optimization in the industry. And also we can have some inventory based QR systems, where inventory and warehousing management becomes more easier than what we are doing now with these QR based systems. And virtual audits and regulatory compliance also can be made to the perfect with this AR VR models. As told previously, these are the AR tools that we can use, like Unity. We know, like most of the gaming companies use this Unity model, and we can combine Unity with Euphoria, and we can have Jigspace, JabWorks, ARKit, and CoSpace Edus, and which are mostly platforms which are available open access. So we can use these platforms to train the students, and we can get things out of this. So this is how a simple layout on how we can create a AR model. So it's as easy as we do things on a computer video editing thing. So like first thing is we need to know what is the basic layout of a plant, and we can use that layout to create a 3D models. Today we have various tools again which can convert a basic 2D plan into a 3D plan. Like we have Unity 3D, we can use SketchUp, and all these things can make a 3D model. And you can use that 3D model, and you can import that 3D model into various AR platforms that we have discussed previously. So you can use Zigspace or ZapWorks, and you can upload that, or you can import that 3D model into that, and automatically with the technology we have today, we can have very interactive VR space we can develop. So it's a simple plan development that we can have as an course output, or as an mini project we can give for the students, and that is how we can train them in this technology. So examples, maybe we can have AR based clean room walkout, or tablet compression mission visualizations, or we can have something, or we can do this with this we have discussed in the previous slide. So next important thing is smart maintenance and SOP training using AR. So today SOP in the era of 3.0 and 2.0, we used to have a lot of physical SOPs and procedures for the maintenance of the labs and equipments. But identify equipment, view maintenance instructions, and all participating, we can update them using the technology, and people who goes to the equipment or the machine can easily understand what the possible troubleshoots. And also we can access digital SOPs in the real time using this AR technologies. So AR based SOP training, we can teach demonstration for the students using the missions. When students scan the missions or QR codes or equipment labels. So simply if they can scan the mission, they can get the SOPs, they can get the safety and precautions and all these things. And this is how industry is working in the 4.0 era. And this is what we can do exactly in the labs if we can train our students on the tools that we have discussed. So classroom activities for this, maybe you can give activities like you can ask the students to create a QR code and place them on tablet compression mission or whatever the mission you have. And you can ask the students to label the parts of the mission or something else, or troubleshoot guiding. Or you can you can ask the students to create a mini project like QR instructions for the cold stain storage. So for the vaccines and all these things, they can ask the patient to create some mini activities using this VR activity. So this is how we can move ahead by making the students to learn more about ER VR, and this is how it is anticipated that we can go ahead in the era of Pharma 4.0. With this, I close my presentations, and thank you all for your patience. Thank you.
Thank you, Dr. Babiraj, for providing valuable information related to the AR/VR tools, which was included in the current PCA syllabus. You given brief information related to the AR/VR tools. Thank you. The next session is by Dr. N. Sylvia, Madam, related to the AI tools in natural drug discovery process. Madam, start the session.
Good morning, everyone. This is Dr. N. Sylvia from Department of Pharmacognosy. Welcome you all to the FDP program on AI and data analytics in pharma curriculum. Today, I am going to present the artificial intelligence in natural products, drug discovery, and pharmacognostic model. So, in this, I would like to cover the following one. So, with regard to our new PCA syllabus. So, what are the new subjects that are being added in the new PCA syllabus with respect to the pharmacognosy subject and department, and overview of the syllabus and the various techniques that are used in the natural product drug discovery, and role of AI in the natural product discovery, various techniques of AI, and various data sets for the pharmacognostic studies, various modeling approaches, and the data sources for the natural product drug discovery, databases and AI platforms, and how we can search these data in the various databases. So in this, there are three: plant search databases, traditional medicine drug and formulation search, and phytochemical search, and remaining other databases by which you can search for the information regarding the not only the natural products but also the synthetic compounds also. And lastly, the challenges and limitations of AI with respect to our natural product drug discovery. So these are the subjects which are present in the whole syllabus, a total of 4 theory subjects and 3 practicals with 1 elective. So the previous ones are the pharmacognosy and phytochemistry 1 along with practical, and pharmacognosy 2 with practical, and herbal drug technology along with the practical, and 1 elective as the quality control and standardization of records. This is with regard to the whole syllabus. So coming to the proposed NEP syllabus for the pharmacy. So the total of 9 theory subjects and 4 practicals. Theory are 5 along with 4 practicals, and electives 4. So these are the topics. So in first semester, 1 subject, second semester 1, fourth semester 1, fifth semester 1. All these we are having a practical sessions. And in sixth semester, there is only 1 theory subjects. And along with these are the entirely the cognition subjects. So other subjects they are also having the incorporation of cognition related topics. So those are in the 601T named as the advanced pharmacognosy, and 604T AI applications in pharmaceutical sciences. So apart from this, the electives which is covering the pharmacognosy topics, that is in third semester, sixth semester, and eighth semester, and the last one, herbal cosmetics for the healthcare. This is also an elective. So the new topics regarding to the individual subjects in the first semester. So this is somewhat similar to our old topic of pharmacognosy 1, but the added topics are historical milestones, various herbal pharmacopoeias, ecopharmacognosy, and physicochemical parameters. This we will study in the old syllabus as an quality control as the evaluation as the physical evaluation. But here he has mentioned clearly the what are the parameters to be explained in detail. So next one added is the DNA barcoding, traditional systems of medicines. In the old syllabus we studied only the ISM and H, and present syllabus we has added the Ayush and TCM, and also the phytotherapeutic agents. Second one, the practical related to the first semester subject is the morphological study of medicinal plants, organoleptic evaluation and powder microscopic characters, and in addition to this, they have added the preparation of herbaria, which is not present in the previous syllabuses, either the immediate one or the previous ones. So this requires the field visits for the collection and preparation of the herbariums. In the second semester, so that pharmacognosy and phytochemistry, so the topics that are added are the pathway prediction tools. So we have studied only the biogenetic pathways, but there in the old syllabus, the tools are not included. Here the tools are being included. The second part is the secondary metabolites. He has given a vast number in the previous ones. In the present one, he has added the pharmacognostic study, which is not present in the previous one. In addition to this, he has also added the fingerprinting of medicinal plants using the TLC and HPTLC and types and significance of markers for the phytochemical analysis. So in the second semester, the second subject is the practical. He has included the, as usual, the chemical identification test for the identification of crude drugs, transfer sections and powder microscopies, isolation of active ingredient, and the additional topic that is being added is the experiential learning based experiments involving the evaluation and comparison of field and marketed herbal raw materials. So this is the new added ones in the second subject in the second semester practice. So coming to the third subject, that is the HDT, herbal drug technology. So there is a vast number of addition of syllabus in addition to the present existing ones. Most of the syllabus was replaced by the new things. So we have studied the plant tissue culture. The previous ones, in addition to that, the additional ones that are added is the role of elicitation, genetic transformation, biotransformation, and precursor feeding, and somoclonal variations in the biomass and secondary metabolite productions. So second topic is the biomanufacturing, including the medicinal plant-based biomanufacturing and utilizing the plant factors in obtaining the various valuable secondary metabolites. Third one is the optimization and production of standardized extracts of medicinal products or medicinal plants. So using the analysis of metabolites through the TLC fingerprints and herbal formulations. The previous ones we have added only the phytosol. Here there is addition of liposomes and nano formulations also. Quality control and the standardization of herbal medicines. Previously we had only the WHO. Now they have added the Ayush and EU guidelines also. So in addition to that, the DNA fingerprinting technology and various markers are also included. In the same one, forensic pharmacognosy, an additional topic and advanced topic is also added. The next one is the interactions, either the herb food or the food herb interactions. So in the previous ones, we have studied only a list of food or the herbal materials. So what are those interactions? In addition to that, he has added the cytochrome 450 and the PGP and adverse reactions related to the plants along with the food, which are the allergy, intolerance, and toxicity. So these are the additional topics on the interactions. So coming to the last one, additional topic is the regulatory framework in India, which is for the herbal and ASU medicine. Previously in HCT, we have studied only the Schedule T. Here they have added the Schedule E1 for the poisonous drugs and also the concept of pharmaceuticals and the Ayush Ahara in the Indian traditional knowledge in the HDT subject. So practicals, preparation and standardization of extracts and the evaluation of herbal cosmetics. So these are the similar to the previous syllabus. The additional ones is the survey and the commercial marketed herbal cosmetics and the evaluation as per the pharmacopoeial guidelines. So next one is the industrial pharmacognosy. This is a completely new subject with advanced topics, which is including the sources and the evaluations and the setup of the herbal industries and the institutions and commercial production and standardization of various botanicals and modern methods of analysis of plants and plant-based products, isolation and characterization and commercial production and analysis of bioactive phytoconstituents and international regulatory perspectives. So the practical session in the industrial pharmacognosy including the chromatography based experiments. So previously we have done these experiments in the previous old syllabus topics. Only one or two experiments are being included, but in the industrial pharmacognosy, more number of experiments regarding the chromatography is being added. So isolation and identification is the previous one, similar to that one. So next one is the added one is the preparation of a monograph. So with respect to the listed materials or the listed fruit drugs or the medicinal plants, and standardization of any Ayurvedic formulations, and the preparation and the applications of the folkloric or the region-specific formulations, and lastly the case studies, which are analyzing by the community awareness, their usage patterns and their combinations of the ingredients which are being used by the community in a regular basis. So next one is the advanced pharmacognosy subject. This is also a completely new subject with a very advanced and this is integrating all the departments in the pharmacy. So unit wise they are included all the departments. So in one of the units it is included the pharmacology expertise, including the about the reverse pharmacology and network pharmacology. So analysis inputs will be including the NMR and HRMS. From the chemistry, SAR and PSAR modeling and molecular docking patterns and the studies, their models and techniques are being included. And also inputs are required from the regulatory affairs with regard to the IND, NDA and patents. In addition to that, the cognosy topic or the content is also having included in the advanced pharmacology. So coming to the artificial intelligence in the natural product drug discovery, the various techniques that are being used in the drug discovery from the natural products are artificial intelligence, machine learning, and big data from the natural products, molecular docking, virtual screening and pharmacophore modeling, genomic and transcriptomic tools, high throughput screening, and bioautography. So mainly I am dealing with the first two ones, artificial intelligence and machine learning. So whereas the molecular docking, virtual screening and pharmacophore, and these are being dealt with already with the chemistry persons in the previous days. So a little input from the genomic and transcriptomic tools and also the high throughput screening and the HTS and bioautography. So genomic and transcriptomic tools, genomics are these two tools are being used for the biosynthetic pathways identification and elucidations. So genomics is the study of complete DNA, whereas the transcriptomics is how the genes are being expressed, that is especially the RNA transcripts. The various tools that are being included are indicated as the genomic tools are the DNA sequencing, genome annotation, molecular markers, and comparative genomics. Whereas the transcriptomic tools are RNA sequencing, microarray analysis, and gene expressions. We can get these data from the any biotechnology reference books, or we can have our colleagues from the biotechnology or microbiology departments. With their help, we can deal these two topics in the pharmacology subjects, which are being included as the one or two topics in the one of the pharmacology subjects. So next one is the HTS and bioautography. HPLC, this is an automated method used to test a large number of samples in a short period of time using the robotics or the microplates or the detection systems. Whereas the bioautography is the direct detection of active compounds, plant extracts, and microbial compounds. So comparing the So comparing the traditional approach of natural product drug discovery with the drug discovery by using the AI. So for the AI, the data is being required, which has to be manually supplied for the system or the software. So whereas the traditional approach, this is based upon the ethnopharmacological knowledge, spectroscope-based structural elucidation, bioassay guided fractionation, and this method, the traditional approach is very time-consuming. And even though it is time consuming, sometimes we are with the already discovered compounds. So this occurs because with respect to natural products, there is a more possibility of a compound to be existing in the multiple plants and in multiple geographic regions. So with the data which are being documented or recorded, this data is being utilized by the AI and analyzes this complex biological, chemical, and ethnomechanical data. And based upon this data, it is translating to our input of a new drug molecule discovered. So coming to the role of AI in the natural product discovery. So this is mainly dealing with the 7 parameters. So one is the data mining and knowledge extraction. Here it identifies the medicinal plants and the bioactive compounds. So avoiding the rediscovery by the machine learning, it compares the spectral data and known compounds which are being identical in a rapid manner. Third one is the structural illustration. This is being helped by the AI-assisted interpretation of NMR, mass to the spectral identification of the compounds that are being isolated. Bioactivity prediction, this is based upon the nature of the phytoconstituents, either the molecular docking or the pharmacophore modeling. And we can predict the what is the compound is having the which type of biological activity. And virtual screening of natural products, this is based upon the various libraries and identifying the various binding sites of the targets. D1 over drug design. So this is being generated by the AI from the natural products inspired molecules with the biological relevance and the interactions between the head drugs. This can also be predicted by using the various database driven approaches. So various techniques that are utilizing the AI for the drug discovery are, one is the machine learning. These are the various platforms in the machine learning. Random forest, SVM, KNN. These are useful for the bioactivity prediction and QSAR models and proximity prediction models. So second technique is the deep learning. So this employs the neural networks like the CNN, RNN, GNN, and the applicability of molecular property predictions and also the generation of structures and the interpretation of spectra. Natural language processing, this gathers the information from the traditional medicinal texts and the scientific literature and helps in the ethnopharmacological data mining. Last one is the network and systems biology approaches. This includes the network pharmacology, multi-target drug modeling, and head compound target interaction mapping. So coming to the pharmacognostic data sets, these are being data can be categorized into the various sets like the morphological features, microscopical features, chromatographic data, phytochemical data, spectral data, genomic data, metabolomics data, bioassay data, and ethnobotanical knowledge. Based upon these data sets, the medicinal plants can be screened and the new drug molecules can be isolated from the natural products. Whereas modeling approaches are QSAR modeling technique, chemoinformatics, network pharmacology, omics-driven modeling, and graph-based modeling. So in the QSAR, this is mainly related to the chemical structure, related to the biological activity. Which chemical structure is having the, we can suggest or predict the based upon the structure of the chemical moiety, what is the possessed biological activity will be. Chemoinformatics, this is the molecular descriptions and fingerprints based the similarity and grouping of the natural compounds. Network pharmacology for the multi-target drug action and the head compound gene disease networks. So omics driven is the biosynthetic pathways through the genomics, proteomics, and metabolomics. And the graph-based ones are predicting the activity and optimizing the structures through the graph neural networks known as the GNNs. Whereas data sources in the natural product drug discovery are the natural product databases, chemical structure libraries, biological activity datasets, and traditional medicine records. So based upon these data sources, the machine learning will be interpreting the what are the similarities and differences. Based upon that, it will be giving the inference which medicinal plant will be having the, based upon these properties, what are the phytoconstituents are present and what may be the possible biological activities that can be elicited by that particular medicinal plant or a isolated compound. So these are the databases and the AI platforms by which the data search and mining will be done in the drug discovery scenario. GNPS, known as the Global Natural Products Social Molecular Networking. NP Atlas, traditional medicine databases. So for the chemical search, smiles and the fingerprints are being used. Plant search databases, the databases for the traditional medicinal drugs and their formulations, and databases for the phytochemical search, and other databases which are very specific depending upon the organism or the region or locality. So GNPS, this is mainly the mass spectroscopy ecosystem which is comparing the various spectra, and this will be used for the identification of the either the medicinal plant or the identification of the isolated compound. So this is identifying and discovery throughout the entire life cycle of the data, and from the initial data acquisition to the analysis to for the publication data. So each and every compound is having a unique spectra. So especially the mass spectra. So this having the details about the mass spectra of various constituents of the plant materials. So this is the website link and this is the home page of GNPS. Here we can have the search for the various materials either through the account or through it is a free network database. We can search for the materials. So second one is the NP Atlas. This is a natural product atlas. This is covering the data about the bacterial and fungal. So this is the website link. This is the home page of the natural products. So here we can discover search for the various materials either based upon the name of the organism, that is especially the bacteria or the fungi. So next ones are the traditional medicinal databases. There are so many with regard to the traditional medicine databases, which is the main backbone or the foundation for the any natural product drug discovery. So this is including the TMDL, TKDL, WTMPD, and OSHA. The other ones are the TCM-SP, IMPACT, MEDDB, and TCMID. The first and the last ones are especially related to the Chinese medicines. So this includes the various plant materials and the phytoconstituents that are being especially identified or discovered or researched in the China. So other ones are the related to the worldwide other resources from the traditional medicines are the WHE, TMD, YATCM, TMCTs. So all these are having the gathering the information regarding to the traditional medicines or the folklore medicines. So next one is the SMILES, a simplified molecular input line entry system. So this is widely used for the representing the chemical structures. So this is developed by David Wieninger in 1980s. So here we can have a storehouse of chemical structures for various compounds with regard to their representations of atoms or bonds or chirality in the chemical compound. So next coming to the plant search databases, the first and foremost milestone or the breakthrough is the first we have to identify the plant. So no person is, even the botanist also cannot identify, even though he is a well experienced one. So based upon the data throughout the world, they have set up the free databases by which we can identify the plant in our region, so either through uploading the image or the name or some other parts. So through these 4 platforms, we can identify the search on our own without the help of a botanist. If it is not possible in a nearby area, the botanists are not available, we can directly search in the databases. So plants of the old online, JSTOR Global Plants, Tropicos, and New York Botanical Gardens. Here I am sharing the web links and the home pages. So this is by the data, which is a digitized data across the world. So this is, data is compiled from the past 250 years of the botanical explorations and research. This is the Royal Botanical Garden Q. This is named as the Plants of the Old Online POW, POW. So here we can search the plant material either by the name or the plant part or by the region. Suppose I have given the home page searching for the Datura metal. So after searching this one, we will get the different images of the plant. Here we can see the taxonomic details, images, and other informations. So distribution, synonyms, classifications, and publications, other data with regard to this plant material. Here we can also get the direct image of the particular plant so that we can compare the material with our material which is in our hand, whether the features are similar or not, so that we can come to a conclusion whether the species are the same one or a different one. Second one is the JST or global plants. This is also a digitized compendium of medicinal plant species. Here also you can get the herbariums of the specimens in this side. So this is the home page. So here we can search for the material. After typing the material, we will get this herbarium image of the particular material along with the other details. So it will be giving the various details with regard to the phytoconstituents or location or country or the cultivation, whatever the parameters that are available. This database or the search engine is giving the details about these medicinal plants. Next one is the Tropicos. Here I have given the web page link and also the home page. Here also we can search by the names. We have the specimens either by the image. So after typing the image, we will have the list of materials with that species or a particular genera. So we will be getting the details. So upon selecting the material which we want to get the information, next we will have the herbarium specimens like this. So among them, which species or which geographical region the material is belonging to. If we click on this image, we will get the remaining data of that particular image of that particular region and collected by that particular person. This is with the Tropicus. We can search by the name or also by the image. So next site is the New York Botanical Gardens Steere Herbarium. In short, it is NYBG Steere Herbarium. As the name itself indicating is it is a herbarium. So here we can search the plant material. When we type the plant material, we get the herbarium of the specimen model of the selected plant. Here we can search the type the plant material. There will be given the list of this one. When we click on this image, all the data and the images that are being related to that particular plant, genera or species or region will be displayed. Other traditional medicinal database that's the TKDL. This is Indian origin. Next one is the IMPACT, Indian Medicinal Plants, Phytochemistry and Therapeutics, also from India, and also the Indian Medicinal Plants database. Here, the TKDL, this is in association with the ICMR and CSIR. So this is the website link. So this is the Indian digital repository for the traditional knowledge. So this is being developed or created to protect the ancient knowledge and the traditional knowledge from the biopiracy and unethical patents. Through this only, in HDT we have studied the patenting issues of neem and turmeric. So through these agencies only, they are able to provide the Vedic the traditional texts by which we can claim that neem and turmeric are of Indian origin, so that the US patents are being cancelled by the data provided by these agencies. So this is the home page for the TKDL. Second one is the IMPACT. So this is the second site for the traditional the ethnobotanical usage search. So this is being having the decisive data about the various medicinal plants and their uses. This is all based upon the textual information on the Indian medicine, and also the research articles which are being published, which are containing information about the traditional medicinal or folklore drugs. So this is the site. Here we can browse by the phytochemical associations. Here we can type the name of the medicinal plant or name of the phytochemical or the chemical category of that or class of the particular compound, or we can search by the therapeutic usage in the IMPACT platform. So next one is the Indian Medicinal Plants database. This is the website link and this is the home page. Here we can search by the name of the plant or by the Ayurvedic entity. In addition to the remaining websites and the databases, it is also giving the information about in which shlokas with regard to the Ayurveda, the mention of this drug is being indicated. Here we can have the shloka in the Sanskrit language and also in the translation of this one, and also he will be having the descriptions, and also we can have the, as this is the Indian medicinal plant database, here the plants with the state wise. So what are the medicinal plants that are available or cultivated or a wild ones? The entire list of plants is available in that particular state that are being can be searched in this state. Suppose we want to do a research on a particular plant and we don't know where the plant is available. In that case, if we come to know across whether it is available in a particular area or region, we can search through this website whether it is available in that locality or it is a native of that one or an imported one, so that we can get the materials and procurement of the medicinal plant materials for the research. So this is the Sanskrit or the slokas, Ayurvedic identity and the slokas. So here we can also have the inputs from the Ayurvedic textbooks. Coming to the last one, that is the phytochemical search. Here there are 4 to 5 databases. So first one is the Dr. Duke's Phytochemical and Ethnobotanical Database. Second one is in the KNAP SAC Comprehensive Species Metabolite Relationship Database, and PhytoHub, Coconut and Ocean. So the first one is the Dr. Gives Phytochemical Ethnobotanical Database. This is the website link. So this is database is having the in-depth information about the plant, chemical bioactivity, and ethnobotanical usage of particular search or the plant material. So this is the home page. Here we can search. There may be a normal search and advanced search. So in this, these parameters, either the biological activity, chemical, or ethnobotany of the plant, and the plant name, or any disease associated, or the treatment for which it is being used, and any folkloric uses or the regional uses of that particular plant. By these categories, we can search the information in the Dr. Dukes Phytochemical and Ethnobotanical database. So second one is the KNAP SAT. So this is the comprehensive database having the natural products, chemistry, and metabolomics. Here it is having about 63,000 metabolites and their relationship with the species. This is can be in the search can be done by the organism name or metabolite name or the molecular weight of a particular compound, or we can give the chemical formula for the search in the this database. So this is the home page. So after getting this home page, either here or by clicking this one, we can go to the search page. So after this, clicking this link, so it will be taken to this site. Here, a varied group of search engines or categories are being there. So depending upon that, either the core system, search engine system, or the block clustering. So when we select these ones, we will get the what are the information, which category and which division or area. We will get the information about either the plant name or by a chemical name. Next site is the Phyto Hub. So this is especially for the dietary phytochemicals and their human and metabolites or animal metabolites. After taking the particular material, how it is metabolizing and how it is transforming. So this is the home page of this one. Here also we can search by the material. Especially this is only related to the food, not with respect to the pharmacology or therapeutics. Mainly the majorly is giving the dietary information about various plant materials. So in this, after typing the particular plant material, what are the chemicals that are being present in that particular plant material or a food material? They will be displayed. When we select a particular chemical or compound, so this will be showing as the information with regard to the structure and how many species or how many plants it is available, in which forms it is available, which part it is available. All these data will be given. So here we have to type the plant name. So this plant name will be giving the chemical compounds. After selecting the respective chemical compound, the entire data will be given in the Phyto Hub. Next one is the coconut. This is the collection of open natural products. Here also is mainly with respect to the based upon the molecules. Here we have to give the name of the compound. After typing the name of the compound, so when you go for the search, so this will be giving the information like this. First, it will be giving the possible structures and their linkages and their different 2D or 3D forms. After selecting the particular structure or an image, we will get the information like this. The entire molecular formula and the structure of this particular compound and various species and plant parts and regions where the particular molecule or a chemical entity is being present. And the last one is the phytochemical search is the Oshadi. This is the online structural and analytics. This is based upon the database for the herbs of the India. This is the website link, and this is the home page for the Oshadi. So other databases are the supernatural. So this few of the databases are also included in our syllabus in the 601T and 604T subjects. So this is the supernatural database link, and this is the home page. We can search for the materials that are present with respect to the name of the plant or phytochemical. NAPRALET, this is the database for the natural products, ethnomedicine and pharmacology and botany. This is the website link, and this is the home page. Here we can browse through the various searches. Ayush research portal, the website link, and this is for the mainly the providing the evidence-based data across the Ayush systems. This is especially related to the India. Here we have multiple categories. Here we have to type what the information, the plant material or category or any other fields. So this will be giving the research papers, evidence-based data for the folklore, other traditional medicinal systems. EMNPD, this is mainly for the endophytic microorganisms from the natural organisms. This is the website and this is the home page. Here we can search or browse and we can download the what are the information that is being displayed. So CMNPD, so this is with respect to the marine natural products. This is the website link. This is the home page. Here we can browse and we can type the information regarding to the marine organisms. So Wikidata natural products, this is for the identifying the chemical compounds with respect to the organisms or geographical locations or the literature. So this is the website page. Here we have to search the browser or what material or what plant material the compound we are requiring. So coming to the last one, so we have seen the various databases, search engines, the data, how we can the artificial intelligence and the machine learning is giving the inputs. Even though it is having a vast data, so many websites, but the main drawback is limited or the high quality data because all the data has to be curated manually. Only the human beings has to provide the data, then only the AI and the ML will give the input for us. But preparing the data and providing data is the main task or the main drawback. So because of lack of data and also quality of data, the interpretation will not be an exact one. Whatever the input we will give, based upon that only the output will be like. So second drawback or the limitations are the model interpretability issues. So as this is a machine, not a human, whatever the process or the what are the program we have given, so it will go in that line. But whereas the human brain, it will think on the pros and cons, whether it is a possible one or not. That technique or that logic is not in the machine learning or the AI. And also it needs the, as it is the new emerging era, it needs a validation. That is experimentally it should be validated and such that the results that are being obtained are true or can it be accepted or may be meant for the any corrections. So thank you for your patience listening. So thank you all.
Thank you, madam, for giving in detail information related to the databases which are useful for searching of the plants going to study in pharmacognosy. Thank you, madam. The next session is by Dr. K. Venkateswar Raju garu related to the innovations and entrepreneurship, the role of AI.
వీడియో ఆన్ చేయాలా మరి people. So due to the artificial intelligence and blockchain technology, nowadays RWA tokenization, product passports, actually everything is token. You can verify the genuineness of the token. And product passports, whatever the product you are using, it will be having a unique ID. And decentralized financial these things, all these are playing a increased activity. IoT, Internet of Things. Internet of Things means actually these are the devices that collect and exchange data in real time. And it has revolutionized the way business operate by enabling continuous monitoring, automation, and data-driven decisions. So these entrepreneurs, this presents an opportunity to develop innovative solutions that enhance efficiency, reduce costs, and improve user experience. So IoT, this is the role of IoT. The IoT business had grown exponentially in billions. Sustainable technologies. Next is how to sustain. The technology should be able to sustain itself. Nowadays you are seeing AI requires huge amount of energy in terms of electricity, in terms of cooling. All these things should be sustainable. So next, in entrepreneurship also, generally sustainability is no longer a corporate social responsibility. It is a business model. So social entrepreneurship. Social entrepreneurship is this entrepreneurship also is based on the social impact investing. So the entrepreneurs also are investing where there is use to the society, or it is having an positive impact on the environment. So this is an example. Climeworks. Climeworks means it is a pioneer in the direct air capture technology where CO2 is converted into permanent storage. So here big companies like Microsoft and Shopify, they are fulfilling their CSR initiatives by tie-ups with these environmental focused entrepreneurs. So that is providing a business opportunity to these new startups. Social entrepreneurship, only the profit with the purpose. Nowadays, the people are not only focusing on the profit, but also the purpose also. So pension funds, major institutional issues, these are also moving capital into place-based investing. So where they can solve the local housing, local energy crisis, like that. So AI also is used for good. Startups are using AI to democratize access to legal and financial services in emerging markets. So here you can see the impact investing. Environmental goals, they are allocating 50% of the funds, social equity 30%, governance and ethics 20%. So global startup trends and the rise of the global south. Previously investment only used to see in the US or Silicon Valley, but nowadays that shifted. You can see Middle East, UAE, like places like UAE, there the investment is around $62 billion, and India you can see $51 billion. All this shift is actually flowing where there are real problems and you can solve it through the high talent. So these are the challenges in innovation and entrepreneurship. Future outlook. Here, future of innovation and entrepreneurship will be shaped by the convergence of multiple technologies, including AI, blockchain, and IoT. So whoever is an entrepreneur, he should be having this multidisciplinary approach. He should have all these skills along with business acumen and social awareness. Critical thinking, creativity, and adaptability are needed for the success. So increasing focus on sustainability and social impact will continue to influence business strategies, encouraging the development of responsible and ethical solutions. So here you can see the unit-wise, whatever we are having in the syllabus, it is focusing on the entire entrepreneur's journey. First unit is focused on the foundation of the innovation and ecosystem. Second unit, ideation, how to get new ideas, how to identify the gaps, and how to generate viable concepts. Next is validation. You should be able to validate the idea by having a minimum viable product, whether the product is viable or not. Is your idea having a viability? That is studied in unit 3. And next, operations. Operations, you can see architecture and business models, what are the legal structures and what is the funding. These things you can see in the operations. Next is scalability, where what is the market growth, emerging tech, and what is the future aspects that will be covering in the 5th unit. Next, foundation. So here you can see the 4 engines of innovation. There is the difference between the invention and innovation. Invention means whatever you are creating something new, that is called as invention. It is only focuses on the scientific discovery and technical achievement. But innovation is different. By applying that solution to an actual market gap, that is called as an innovation. Here the focus is on economic growth, job creation, and solving societal problems. First thing is only focusing on creating new things. Next thing is focusing on the applying solutions to actual market gaps. And first thing is focused on the scientific discovery. Next, innovation is focused on the economic growth, job creation, and solving society problems. So these are the 4 engines of innovation. One thing is product, process, business model, and social aspects. Next, startup ecosystem. So here, whenever somebody is starting something new, various multiple stakeholders are there. So who are the stakeholders? What are the issues? You can see here. First thing is support services. Whoever is starting, you should know the financial, legal aspects, and operational infrastructure. Here you can see the central entrepreneurial student. He should have this idea. Support services he will be availing. Next, academia, the foundation of research talent, and he should focus on these things. Next, government support. He should know what are the regulatory frameworks and what are the incentives he can get from the government. Next is incubators. Incubators means nowadays you can see places where the entrepreneur can go and plug in and work, and he can have early access to early resources. That is called as incubator. Next thing is he can have mentors in the industry where they can guide him through the industrial experience and navigation. Next, this is the critical thing, investors, people who can invest money, who can fuel the growth with essential capital. So these are all things are involved in the startup ecosystem. Next, ideation and discovery. Now we have seen in the ideation and discovery how to get new ideas, whether the idea is feasible. These things you should be seeing in the unit 2. First thing, problem identification, broad thing. Next, in that you should be able to generate ideas. Next, opportunity, whether that opportunity is viable or not. All these things you should be able to track. Here are the tools, some things, techniques, observation, empathy, mapping, user interviews. For problem identification, you should have all these things. Next, idea generation. While generating an idea, you can use CAM for mind mapping, design thinking, etc. And opportunity analysis, SWOT analysis, competitive landscape, focus. Then you should focus on the true commercial feasibility. Next, validation. We are seeing in the unit here the validation engine. Here you can see the learning. First thing is he should be able to learn, analyze feedback, making the critical founder's choice. Pivots means whether he can change the direction or further stay on the course. Next, building. Building means he should be able to develop the minimum viable product, utilize tools for rapid prototyping, etc. And next, another thing is the measuring thing, validating against the market, executing the A/B testing, split testing, and structured customer interviews. So, in the 4th unit, you can see the value proposition design, customer relation, channels, customer signals, key activities, key resources, cost structure, and revenue streams. So, unit 4, you can also see the legal matrix. There are various types in the startup ecosystem. Sole proprietorship, only a single owner can do all the things. Partnership, he can share the things with other partners and ownership. And private limited, by using the private limited, he can save himself from legal issues and liability issues and structural complexity. So also these things you should be able to know. IP, patents means he can get the intellectual property rights, trademarks, copyrights, etc. Next, funding. After all these things, you should be able to get the funds. One thing is bootstrapping, self-funding. Angel investors, you can approach other persons, venture capitals, institutional funding, and handrail, strong foundational team and HRD. These are supported. So all these things are needed for a entrepreneur. Next is scaling and the future. Scaling means growth. How to grow your product or prototype into a vast market by using marketing, sales, and aggressive user acquisition and retention strategies. Exit strategies. Exit means rewarding investors through mergers and acquisition. By you can invest or divest. Next is future. What are the disruptions? You should have the latest knowledge of the artificial intelligence, blockchain, IoT, and sustainable technologies. Social entrepreneurs, impact investing, all these things you should have a knowledge. Next, these are the phases of the experiential roadmap. 50% is academic and 50% is experiential. So here you can see phase 1, phase 2, phase 3, and phase 4. Phase 1 awareness, phase 2 action, phase 3 skill building, and phase 4 execution. Founder, here you can see the lean startup methods, 0 to 1. All these things are the models, and so this is the future of healthcare requires founders. So here all these things are included in this B-Pharmacy Fine Art Treaty syllabus, and this is actually launch pad where you can identify the problem, build the minimum viable product, and pitch your vision. All these are included in this syllabus. So this is about the startup ecosystem, and this syllabus is in aligned with the national educational policy, and I think this syllabus will be helpful to guide the entrepreneurs and through their journey for the startup ecosystem. Thank you. That's all.
Thank you, sir, for giving valuable information related to the entrepreneurship and stages of entrepreneurship and AI role in entrepreneurship. With this session, we are completing the 3-day FDP program. After completion of 3-day FDP program, the concluding remarks are this EFDP successfully highlighted the transformative role of artificial intelligence, machine learning, and data analytics across various domains of pharmaceutical sciences and healthcare in alignment with the NEP 2020 and new PCA syllabus. The sessions are emphasized the applications of AI in medicinal and pharmaceutical chemistry, pharmaceutical analysis, biotechnology, genomics, personalized medicine, public health, food waste management, pharmacognostic data modeling, pharmacy automation, and supply chain management. The discussions on innovation, entrepreneurship, and Pharma 4.0 technologies such as AR/VR showcase the importance of interdisciplinary learning, skill development, and technology-driven education. Overall, the program motivated the participants to adopt modern digital approaches for advancing research, industry practices, healthcare delivery, and pharmacy education in the evolving global scenario. We express our sincere gratitude to the management and principal of Sri Vishnu College of Pharmacy for their constant encouragement, valuable guidance, and support in successfully organizing this program. We also thank all the resource persons, faculty members, and participants for their active involvement and contribution towards making this EFDP a meaningful and successful academic event. Thanking you all.